So the other thing is I do need, and we'll see where we get to, and we'll probably make a choice at about 5 minutes to go, whether you would like to spend the time to do your class evaluations rather than talk about the subject matter for the last 5 minutes. These course evaluations are not just useful for me, could also be useful for the, I think, 63 of you who are taking virology next term, or at least some of you who are. So I do try and take these comments into account. And there are a couple of things that I have changed this term that I'm really interested in you giving me some feedback on. The first of those is that change in terms of clicker questions. And then just the general recording and so on and so forth, how useful those things are. One thing that I haven't done in the past, but I'm also thinking about doing, is warning people about this class just by posting all of the suggestions that I've heard from previous years. So just in case people don't look at rateyourprofessor.com, and I don't even know how many chili peppers I have. So with that being said, again, don't forget, be here on time. And I'll leave this up for a little while. So basically, we talked about gene regulation since the last midterm. A couple of people asked me regarding the final, is it going to be cumulative? No, but I will expect you to know the material that we've learned through the first 2 midterms. So nothing specific on that, and it will be exactly the same format as the previous exams. So basically just like a 3rd midterm, 50 multiple choice questions. So we did very briefly talk about protein folding right at the beginning of this section of the course. A lot of this has to do with the chaperones, and this is, of course, just one of the ways that proteins get folded or not folded, as the case may be. And this case is really just protecting against the incorrect interactions that happen during the protein folding process. Most of the time, we talked about this sort of central dogma of biology, certainly in terms of cellular biology, where we start with the DNA and eventually end up with an active protein down here at the bottom, and how each of these steps happen, and particularly how they're regulated, is really what this part of the course has been about. Cloning, I wanted to emphasize that the cloning of Dolly the sheep and various other organisms is not the same as molecular cloning. When we talk about molecular cloning, we're talking about making many copies of a particular nucleic acid sequence, and usually a quite short nucleic acid sequence. If we're talking about cloning from somatic cell nuclear cloning, we're talking about cloning of whole cells and all of the genetic material in one of those particular cells. And the classic example here, and classic being 20 years ago, which is kind of amazing because I remember it quite clearly, when the sheep were originally cloned. And the basic message here is, again, we just take a nucleus from one cell, put it into another cell, and magic happens. And in very few cases, this is very low efficiency, you'll actually end up with an organism that had the genetic information of that nuclear donor. One of the things that I think I didn't do a terribly good job of explaining the first time round, so I give myself a second chance, another reason to do these review lectures, has to do with not so much the outcome of this slide, which is supposed to show differential gene regulation in different kinds of cells with different kinds of genes, but really a lot about the technique which is used for this, called RNA sequencing or RNA-seq. And the idea here is that you get cells, usually populations of cells, and then we're now getting to the point that you can do this with single cells, which is totally cool technology that we haven't had a chance to talk about. Hopefully, Dr. Bartlett will talk about it next term in recombinant DNA. And that is to look at the RNAs which are being made in any particular cell. and looking at absolutely all of those RNAs, again, in usually this case a bulk kind of scenario. So you get lots and lots and lots of short sequences of RNA, and then because we know the genome sequences, you can pile up those sequences and say where they are. And so if you look, and again, we'll just talk about the top part here. This is looking at the beta-actin gene. Here, for every RNA that you see, you'll put a little dot. And if there's the same RNA which is there, you'll put a dot on top of it, et cetera. So you end up with a bar if you have a lot of RNA with that particular sequence in it. This is a great way to tell where you've got exons versus introns, because you have a lot fewer RNAs in these introns than you have in the exons. But also you have these untranslated regions, 5' untranslated regions and 3' untranslated regions, although here you can't see it really that clearly. And the idea there is that in your final messenger RNA, it's not just the exons, but you have the extra pieces at either end as well, which turn out to be very important for gene regulation purposes, particularly a little bit further downstream in the process. Now, when I say downstream in the process, it's not relative to the promoter where you're getting transcription. This is now more of an information transfer process downstream between the DNA that you start with and that final active protein. Again, the first thing that happens here is control of transcription. Why? It's really because that's the most energy efficient way to control any particular gene, but it also takes some time, and that's why you can have these wonderful circuits that we'll talk about in a couple of minutes now. So the transcriptional control really seems to be where the vast majority of gene expression regulation takes place, and that's why we spent so much time talking about it in this class. But definitely, before you get to a final active protein, we have RNA processing control. The main thing that we talked about here has to do with alternative splicing. So you can get differential kinds of splicing, but there's also the processes of capping and tailing, which are going to be different, and even some RNA editing, which we'll talk about again here a little bit later on. One of the things that I talked very briefly about is where the RNA ends up in the cell. That's very important for developmental biology purposes, and this really very often has to do with that 3' UTR sequence, and we'll take a little bit more look at that later on. How stable your messenger RNA is can determine how much protein you have, how much it gets translated, and then finally, this activity control, the very last step in terms of activity of a protein or lack of an active protein. This, of course, has happened way after you've made everything, so it's not terribly energetically efficient if you want to make a major change, but it's a very fast way of changing. And so we talked about signaling pathways, and a lot of these signaling pathways will have phosphorylation or GTPase activity or something like that, which is a very rapid kind of response that happens. But most of the regulation is happening here at the transcriptional level. So transcription, of course, is going from our friend DNA to RNA. How is that being regulated? It's the proteins which are making the RNA, and how you get those proteins that are making the RNA to the right place. And this is all about promoters, again, from the last section of the course. Getting the RNA polymerase to the appropriate place to start. And once you've done that, again, the majority of the time, if that polymerase is sitting there, you will get transcription. As always, this is biology, so we have exceptions to that particular rule. So how do most proteins interact with DNA? They're going to mostly interact in the major groove of DNA. So here, the major groove of DNA. And very often, it's going to be one major groove, and then the next major groove, one turn of the helix separated from each of these. And this is very often dimers, and we'll talk about some of those dimers again here in just a second. So these DNA binding proteins interact major groove DNA. It's because that's where we have all the chemical information, all your hydrogen bond donors, hydrogen bond acceptors, methyl groups, etc. And then these are going to be your cis-regulatory sequences. And so what's a cis-regulatory sequence? Cis-regulatory sequence just means it's on the same piece of DNA and near to the promoter that it's regulating. Now, what do we mean by near? In the case of bacteria, it's probably going to be tens to hundreds of nucleotides away. In eukaryotic cells, it can be tens of thousands of nucleotides away. How do we bind to DNA? In bacteria, the vast majority of proteins are the helix-turn-helix containing proteins. Helix-turn-helix is a DNA binding motif. The helix-turn-helix in and of itself is not a stable structure, so it's not a domain. It's just a motif. And that's these blue and red helices that we have here. Blue helix, which is the helix, followed by the turn, followed by the helix. And these fit into adjacent major grooves of DNA. And this 3.4 nanometers, I went back and checked, is actually correct. I misspoke in one of my lectures. Between these two is because it's 10 base pairs per turn, again, from Watson and Crick's structure. And 0.34 nanometers is the distance of one base pair, so 3.4 is 10 bases. So in regular B-form DNA, 10 bases, you're going to get back to the same side and have that same major groove in that place, which means that when you have one of these helix-turn-helix and typically a dimeric protein, each of these, again, is going to bind in adjacent major grooves of the DNA. And that brings me to probably the slide that I've gotten the most questions about in office hours, which is this one. Dimeric symmetry, dyad symmetry, half sites, etc. The idea here, again, we're binding in the major groove, but what the heck is going on? What's the DNA binding? What's not DNA binding? So I tried to mash these two images together. And I usually say I don't put any new slides into my review lectures. I'm breaking my own rules here. But the idea is that here we have our recognition helix. It represents, I call this the swoosh of your dimeric protein here. Here's another recognition helix in your dimer. It's recognizing this piece, and it's really sitting in the major groove of DNA here. In the background, where these two are touching each other, that's the dimerization. And so that's these two beta strands here, which are interacting with each other, that are not interacting with the DNA. Does this kind of sort of make more sense now? Yes, no, maybe. So the beta strands are just holding each of the monomers together so that the HTH can sit in adjacent major grooves. And because they are identical alpha helices, they're going to be interacting with identical sequences, but they're in inverse orientation. And so that gets back to our dyad symmetry here, where if you look at the sequence, it's AACAC from 5' to 3', and in the opposite strand, AACAC from 5' to 3', because it's the same alpha helix. It's a homodimer, which is going to be interacting with these sites. Yeah, Olivia. So the question is, can those beta strands dissociate and then reassociate? In the case of CRO, this particular protein, that's very rare that they do that. Actually, the individual monomers in this case are not stable. There are other examples which is a great lead-in to this one, where you can actually mix and match those kinds of things. But the actual presence of those beta strands, those two short beta strands, they're going to be incredibly specific in terms of how they interact with each other. So you've got to have the exact right length of beta strand, the exact amino acids to have those kinds of interactions. And these more general dimerization motifs that I'm just about to talk about are then ones that multiple proteins share. Is that a question? Was that sort of your question? Okay. So the idea here with dimerization motifs, and interestingly enough, again, getting back to your question, Olivia, if you look at heterodimers in bacteria, you actually don't see them very often. You almost always see homodimers. And that's probably because the genome is a lot smaller in many of these cases. And so the specificity issue is not such an issue. So, but we'll get to that in just a second. So dimerization motifs, and a great point in sort of, I think, leading into that, is that you actually can, in many cases, and this is particularly true for eukaryotic DNA-binding proteins, mix and match different monomers to get dimers. Now, how does that mixing and matching take place? The vast majority of those are really two basic kinds of motifs. There, I shouldn't say basic because that's something else, but two fundamental motifs that you see a lot for dimerization. One is the leucine zipper, and all that is two alpha helices with every 7 amino acids a large hydrophobic amino acid side chain. And this is then very different than what you see in Crow. You've got very specific amino acids which are there. Here, you can have large hydrophobic amino acids, usually leucine, but it could be lots of other ones as well. And so just because of that, you have a dimerization which can happen due to interactions of these two helices. Now, this dimerization, and it turns out that these alpha helices wrapping around each other, doesn't have to do necessarily with DNA binding proteins at all. There are lots of leucine zippers that have nothing to do with DNA binding proteins. Sometimes you want to have two proteins that come together for other reasons. But if you happen to have a basic alpha helix, which is right next to your leucine zipper, this can now bind to DNA in a way a lot like scissors would if you open up a pair of scissors. And so that will interact. So the dimerization domain is not the DNA interaction domain. The leucine zipper is a dimerization. It's only if it's got a basic helix that is directly adjacent to it that it'll be a DNA binding protein. Yeah?
So the dimerization interface is more specific than where it's interacting with the DNA? Yes or no? Because these ones can be the homodimers or heterodimers, correct? Or
Okay, I'm not sure I quite understand the question. I guess, so I'll try and paraphrase it. But is the dimerization itself, that dimerization domain, is that going to be more specific than the DNA binding part of it?
I guess I'm just confused on what the difference is between where the leucine zipper is attaching to the major group and the dimerization interface portion. Is it two separate portions? Or
Okay,
is it all one strand?
so it's really two separate portions. Here it's shown as one continuous alpha helix, but you can clearly separate the two. Sorry, the question here really has to do, I think, and correct me if I'm wrong here, with basically how can you separate that dimerization from the DNA binding? And can you do that in such a way that you have different specificities potentially? So that's, again, this is a paraphrase of that question. Yeah, and so yes, you can. And so if you've got again, then these leucine zippers, all they have to have is large hydrophobic amino acids every 7. The DNA binding has to be much more specific. So it's going to be interacting with some kind of base pair in the major group of DNA. And so that's how you can get the mixing and matching is really happening with your leucine zipper. And you really can separate those two from each other. It turns out that in most of these proteins, it's one long alpha helix. And so that's where I think some of the confusion comes in.
Well, and I remember you saying in the actual lecture that that the upper portion is in heterodimers it doesn't really know what's going on down below, and so that was kind of what was going on.
Yeah, and so again, this is another way of thinking about, and this has to do with actually one of the slides I wasn't going to show, which is the modular state of these proteins. And so you can have a dimerization domain and a DNA binding domain, which can be completely separable from each other.
Okay, I think that might be right.
Yeah, your question.
Yeah, the figure doesn't show the basic alpha helix that you're referring to make it a DNA binding.
Yes, so in this particular figure, and this is one of the arguments I have with this textbook, and practically all molecular biology textbooks actually, so it's not just this one, which it doesn't show that basic helix. And so a lot of times, and if you just look at this in the literature, it will be a B-zip protein. So the B stands for the basic helix, and then the zipper is that leucine zipper, or BHLH. So basic helix loop helix. And again, this helix loop helix is just another way of getting your dimerization to form. But it's the basic helix which is interacting with the DNA.
So you're saying that those two have to be basic, or is there a third helix that comes in?
Okay, yeah, so thank you. So basically, where's the basic helix? I'm just paraphrasing here. The basic helix is everything from here on down. So that's your basic helix, that's a basic helix. Over here, this is your basic helix, that's your basic helix.
And then they're base pairing, they need to be basic because of the negative charge DNA.
So they are negative because they're interacting with the DNA. And basic, yeah, so okay, basic is going to have positive charge, so it's going to be interacting with the phosphate backbone. But you also have side chains which are going to be interacting specifically with the base pairs. These are specific, and it's not just a basic helix. It's a basic helix with very specific amino acids that are partially basic and partially non-basic, which are going to be interacting with the bases.
And that has to do with the alpha helix because they stick out.
Yes, and so in your alpha helix, you're exactly right. The amino acids are sticking out.
Okay, awesome.
Yeah, Evan.
So the dimerization, that portion is about getting the part that's going to interact with the major group into the correct place.
Exactly. So the dimerization, all that it's doing is getting those basic helices into the right place to interact with DNA. Yes, no, better, good. Okay, so why dimers in the first place? Couple of reasons. One, of course, is that you can heterodimerize, and so you have more possibilities in terms of how you can interact with your DNA, because each of those half sites, so each of those individual basic helices, could be interacting with a different sequence. And so now you have more possibilities. Also, each of those alpha helices is usually going to interact with 5 to 6 base pairs. 5 to 6 base pairs, particularly in a large genome like ours, is not going to be enough specificity. So you need those two to get high enough specificity, and that would even be in the case of a homodimer. You need to have that binding specificity. And then, of course, you can also have the cooperative binding, where you have much more of a switch activity as you raise protein concentrations. If you just have a single protein binding as you raise protein concentrations, it's going to be a exponential, actually asymptotic rise to where you have binding, whereas it'll be a sigmoidal one if you have cooperative binding. How do we know about binding? A couple of techniques. Again, this is where lots of people have been asking me questions in office hours. Reminds me again, for those of you who haven't filled out your course evaluations yet, let me know if you think I should talk about methods earlier on in the course. I think I should, but it's too late this term. So these electrophoretic mobility shift assays are literally looking for a protein that's binding to a sequence of DNA. So you have a particular sequence of DNA. You are trying to find a protein that's associated with that particular sequence of DNA. How do you know that it's associated with that sequence of DNA? You do DNA gel electrophoresis. Another reason I'd like to talk more about gel electrophoresis earlier on in the course. That DNA gel electrophoresis is separating DNA. If there's a protein that's associated with that DNA, it's going to be larger. So it's not going to go through the gel as quickly. And so this is a case where you have multiple proteins that are bound. Much more frequently, you have something like we have down here in the lower right, where we have one protein which is bound, in this case the C2 protein, which is bound to DNA. And the reason that you can tell it's bound to DNA is because the mobility of that DNA has changed. So this is finding that you have a particular DNA binding protein. This is potentially what you would use, I think in last time we asked about the little blue bumps that you had in some of the chromatographies. This would be a really good way of saying, oh, okay, I'm finding a protein in this fraction in my chromatography which binds to this particular piece of DNA. So you're using it as a way of testing each of those particular proteins. And in fact, this is exactly how we got TF2A, B, C, D, E, and F. All of them were different fractions that came off of these columns that bound to promoter sequences in eukaryotic promoters. Yeah, Olivia.
But this isn't very specific, right? Because you're getting like multiple proteins rather than just maybe the one you want to isolate.
So the question has to do with how specific is this? And this is why I also don't like this example terribly well. Usually you're going to be finding one or a few proteins that are associated with a particular piece of DNA. So say you have one of those sequences that has a diad-symmetric sequence in it that you found by doing your DNA sequencing. You think you want to try and find a protein that's associated with that. You have that as your DNA that you're going to detect, and then you put various different fractions into it and try and find one that causes a shift. Now, this case is a whole mixture of different ones, but you can separate those now, and this is literally chromatography at the top here. This is why we have this green line. It's different fractions, and each of those different fractions has a different protein, which is binding to all this same DNA, and so it's probably a pretty big piece of DNA, probably a promoter sequence, but those are separable proteins. Yeah, Damon, you have a question? Yeah.
So are they sort of, is it actually DNA affinity chromatography and then EMSA and then the DNA footprinting in terms of increasing specificity? What I wrote down was that you had DNA protein that we were identifying as affinity chromatography, and then you're identifying the specific protein that's binding to EMSA.
So the EMS is a way of looking at what you have. And so you'll do an electrophoretic mobility shift assay on any given fraction that you get off of a column. So this is a way of determining whether, again, it's this little blue region. And so you'll take each one of those fractions that you've gotten from some purification experiment and test to see if it's binding to DNA. So this is a test to see if it's binding to DNA. Okay, so getting to your point, however, about the DNA affinity chromatography, is once you've found a piece of DNA that will get shifted by some fraction, then you could use that DNA sequence that you found to try and purify that protein. And that would be the affinity chromatography. Here, again, a DNA affinity chromatography. Just like the other chromatographies that we talked about, I guess, Monday, where you have a column, you have beads. On those beads, you have something that's going to slow down the progression of a particular protein that you're looking for. Could be because of the charge, could be because of the size, and in this case, because of a particular DNA sequence that it's associated with. Another one of these assays that we talked about is DNA footprinting. Once you have your purified protein, now you want to know exactly what piece of DNA it binds to and literally where on that DNA it's bound to. So you've figured out, probably by doing an electrophoretic mobility shift assay, that it binds somewhere in 100 base pairs of DNA. Now you want to figure out exactly where it's associating with the DNA. And to do that, you do one of these footprinting experiments. And the footprinting experiment means that you've got lots and lots of DNAs up here that have a label at one end. How do we put the label on? Check the box. Check the box so you get the label on it. Yeah. When you're buying your oligonucleotide in the first place. Or you use terminal transferase, or you use polynucleotide kinase to put a label on the end of your DNA. So now you can detect it. And that's what all these black bars down here at the bottom represent, is each of those. And each of these black bars, because it's gel electrophoresis, represents a DNA of a particular length that has a label on it. So if you have this DNA that's labeled, you chop it into little pieces, and in the absence of protein, you'll get lots and lots of pieces, some of which are long at the bottom. That's going to be the top of the gel. Some of which are short over here. That's going to be what you have at the bottom of the gel. If you have a protein that you put in here, it will stop the chopping up of your DNA in this particular region right here, because it can't get chopped up anymore because this protein's in the way. And what that means is you're going to have all of these short DNAs, which are over here, and a bunch of long DNAs, which are over here, and then a gap. And this gap is because the protein has protected the DNA from getting chopped up in that particular region. And literally, you can go through those individual bases and say, this binds between base 438 and 503 or something like that. Yeah?
So if we wanted to experiment and find out what like helix turn helix protein, or at least where it binds on, is this the kind of thing that we would do for that? I'm just trying to make like a whole picture.
Yeah, so you've purified a helix turn helix containing protein. Now you want to see, and you figured out that it binds to DNA in a bigger piece of DNA with one of these electrophoretic mobility shift assays. Now you want to know, does it bind to CATG or does it bind to AAGT, something like that. And so you have that sequence in your DNA, you add it together, you do this kind of experiment, and then you'll see exactly where that is. Yeah?
So with the homodimer association, since they're only bound together through that beta strand association, is it just something that by looking at the sequences and seeing that they're reverse of each other that you know that they bind that way? Or would the chopping up still chop up those two, you know what I mean?
Okay, so I guess I'll paraphrase your question here, if I may. But the question is, if you just look at the DNA sequence, can you predict where it's going to bind to? And is it always going to be like that? Because you have these inverted repeat sequences or something like that.
Rather, like when you have two proteins that come together and make this dimerization happen, but there's space between the DNA, like how that gap works. If you have two gaps, and then you realize, oh, these are two that are linked together.
Okay, so
How do you still come in and cut those two apart?
yeah, so the question here, okay, so I'm sorry, I misunderstood, I think, your question. But if you did one of these footprinting experiments with a dimer, would you see two areas which were protected and then one area in the middle which wasn't? It depends on how you do the experiment. If it's a really high-resolution experiment, like they did here, and I'm not going to talk about all the details on how it did it, yes, you would see two separate pieces. You'd see two areas where you wouldn't have that, and like a footprint in two different footprints. And that's often, in fact, what you see, things like this. Okay, if you have a DNA binding protein, however, and you have absolutely no idea what it binds to, you haven't figured out a gel mobility shift, you haven't found a piece of DNA, you found this wonderful sequence in the genome of the new organism which you've just sequenced that is causing some kind of nasty disease and chews away at legs, whatever it is. And you've done the genome sequence and you say, hey, I've got this protein that looks like it's got a helix turn helix in it. What the heck does it bind to? No idea. So that's when you would do one of these CellX experiments. You have the protein, but you have absolutely no idea what it binds to. So now you take a mixture of all of the possible DNA sequences that you can click on the box and order. So then, once you've got all of these DNA sequences, you clicked on the box in the order, and you also clicked in the other box that said label them, please. And now you can find all of those sequences which associate with your favorite protein. Often this will be by a gel mobility shift assay, because you'll see, okay, all of these different DNAs, all different sequences, there'll be a few that have a shifted mobility because of the protein which is there. Then you take that DNA out and you sequence it and you say, oh, I've got a sequence now which is binding to my particular protein. So, and then you would probably want to do some of these DNA footprint experiments to say, yeah, is it really doing that? Or you go back to the genome of your flesh-eating bug and say, hey, you know, this sequence is here, maybe there's something going on there. Yeah? This can be done for any DNA binding protein at all. And in fact, it can even be done for designed DNA binding proteins that have nothing to do with bacteria or archaea or eukaryotes. And in fact, this is what people do when they're doing those kinds of design experiments. Yeah, Damon?
And none of these things thus far we've talked about just now can be done in the cell itself. They have to be done with the protein and DNA pieces.
Correct. If you want to do it in the cell, you do this. Chromatin immunoprecipitation. So these are much, much, much harder experiments to do, which is why people only end up doing them once you have a pretty good handle on what you think is going on with your particular DNA binding protein. you have your sequence, which you've discovered from the purified DNA. This is great and wonderful with purified DNA, but cells aren't purified DNA. So what's actually going on in a cell, and particularly at any given time? And we've talked about this, and we'll look further on now, particularly having to do with chromatin rearrangements and so on and so forth. Where do you actually have that particular DNA binding protein associated with your DNA? And so the way that you do this, you have your cell under your particular conditions, and as quickly as possible you stop anything going on with the cell and you cross-link your favorite protein to the DNA. You chop up the DNA, actually very similar to what you do with the footprinting experiment, only now you don't have a labeled DNA, you use the protein to fish out those particular DNA sequences. And the way you do that fishing is with an antibody. And so that's the immunoprecipitation part, is that antibody will fish out the protein and any DNA that's associated with it. And then that particular DNA, you can, in the original experiments, people did this chromatin immunoprecipitation and then would hybridize any of that DNA to an array that had all of the DNA of your cell on a particular array. And we didn't talk too much about arrays, but the idea with an array is you have a microscope slide, standard microscope slide, that has 20,000 dots of different gene sequences on it. And then you try and find which of those sequences that you did with your chromatin immunoprecipitation, which one does it hybridize to? And you can say it's this gene or that gene. It's now become much easier to sequence whatever DNA it is that is associated with your protein. So pretty much everyone does the sequencing, and then you go back and look at exactly that same kind of experiment that we talked about a couple minutes ago, which is the RNA-seq experiment. For every one of those DNA sequences that matches somewhere in the genome, you put a dot, and then you get another sequence, you put another dot there, you get another sequence, you put another dot there, and then just see where you have large amounts of sequences that are present inside your genome. Okay, methods, DNA binding protein methods. More questions on those. Okay, let's finally talk about regulation. The classic example of regulation is the lac operon that Jacob and Monod got their Nobel Prize for, even though they didn't know what was going on, which had to do, and this is, all that they knew at the time was the lac repressor. So you have a protein that binds to DNA, blocks the binding of the RNA polymerase. This is classic negative regulation. So binding of a protein that blocks the association of your transcriptional machinery. Of course, what was found out later is there was also, even though there's no such thing as positive regulation, the lac operon did have positive regulation. And the positive regulation in this sense was a DNA binding protein that would stimulate the binding of the RNA polymerase. So both kinds of regulation here going on at one promoter. Questions on lac operon? Okay, why do we spend all this time talking about bacterial gene regulation? Bacteria are very important for disease. They're probably also really important for health in terms of our microbiome. But we really care about eukaryotic gene regulation because that's what all the rest of our cells are doing. But very, very similar processes. Positive regulation, negative regulation, blocking the binding of certain proteins, stimulating the binding of certain proteins, happening through slightly different ways, and combinations of all of these different positives and negatives. And it's not nice and simple like in the case of the lac operon where you have one positive regulator and one negative regulator. Now you've got lots of positive regulators. You've got lots of negative regulators. And how they're all interacting with each other and interacting with the polymerase is going to determine whether you have transcription at one particular promoter or not. And it's not just the DNA binding proteins. We also have non-DNA binding proteins that are very important for transcriptional regulation. These are the co-blanks, the co-activator, or the co-repressor. Non-specific DNA binding proteins. They'll either be just protein-protein interactions, or they could be a non-specific DNA binding protein that will then be brought to a particular place due to protein-protein interactions. And these will change then whatever is happening to the promoter complex. A lot of these co-regulators, So coactivators or co-repressors are going to be involved in changing the state of the chromatin, which is near the promoter or near where those first DNA binding proteins are associated with the DNA. And this is a quick aside I forgot to remind us all here, is that what is the DNA sequence called that an activator binds to in a eukaryotic system? An enhancer, exactly. And what is that DNA sequence called if a repressor binds to it? A silencer, exactly. So repressors and silencers here. And again, 90% plus of eukaryotic promoters will have not just the DNA binding specific transcriptional activator, they'll have something else which is associated with them, again, usually coactivators or co-repressors, and many of these are going to modify your chromatin in some way. Either a chromatin remodeling complex, which is ATP dependent, moving nucleosomes around, or you can also have specific, histone modifying enzymes, and these are the ones that I think are the most straightforward in terms of thinking about if you have a histone acetyltransferase, what does that do? It acetylates the side chains on histone tails. The histone methyltransferase is going to be methylating them. Usually, though, adding acetyl groups is going to stimulate transcription. Adding methyl groups is going to be silencing your transcription. And very often, these are going to be those coactivators or co-repressors. You also have the non-cos. These are the repressors which are going to act directly. And this is the repressors in this case, and this is the eukaryotic system, act either very similarly to where you have the repressors in bacterial systems. It's literally binding of one protein that doesn't allow the binding of another protein. And so the repressor blocks the binding of an activator. Could be interacting because, as we talked about before, many of these activator proteins are sticky in terms of their activation domain, or activation surface as it shows here, but usually this will be a separate domain. This can be blocked in such a way that this activator now can't interact with the polymerase, and sometimes they'll bind directly to proteins which are present there. We already talked about these indirect ones, so I'll skip over that. Wanted to mention now this as the example, even skipped. What everyone loves the most about this course, I think, is the whole even skip gene regulation. And actually, second slide, which I made a mistake on before and have now modified slightly. I will leave it to you to figure out where that modification was. Great to study for the exam here. But in this case, even skipped is an example which we use because it's a relatively straightforward example where we're just looking at activators and repressors, and the presence of activators and repressors at particular places in the Drosophila embryo will lead to gene expression or not gene expression in a particular location in the Drosophila embryo. And so that is here in a particular stripe. This is only one of 7 stripes that forms. I need to get a bigger pointer here so I can see it myself. Right here in the middle, this at the top here is what the regulatory sequence should look like. Now has all these activators associated with it. The mushroom, I'm not going to expect you to remember these things, and the little comma. Mushroom is hunchback, the comma is bicoid. We have these activators. You only get expression regulated by this sequence if you have these 2 activators here. If you have repressors, they're going to block the binding of these activators. Yeah?
That's only for stripe 2, so stripe 3, it might be a different combination of activation repressors?
So exactly right. This is just for stripe 2. For stripe 3, you will have a different combination of activators and repressors because you don't have any hunchback and bicoid left by the time you get to stripe 3, which will be slightly posterior to this. So it will be different regulators which are involved in that in stripe 3 and also involved in stripe 1, both ways. So this is just what's going on for stripe 2. Other questions on this? Yeast mating type switching, I did post an extra reading on this, so the previous version of the textbook. I really like this example. Not so much for the transcriptional regulation, which is relatively boring. It's transcriptional repressors that will repress either the A-specific genes if you're alpha or the alpha-specific genes if you're A. But much more so how you get this mating type switching that takes place. Has to do with endonuclease cleavage and homologous recombination, which is switching mating types from A to alpha. Where you get that information from is in otherwise silenced parts of the chromosome. So the silent cassettes are what gets used to copy and replace the DNA, which is in the mating type locus right in the middle. And how do we have silencing? That silencing is due to heterochromatin formation, and the regions where you have these silent loci are blocked from the rest of the chromosome by boundary elements, which are bound to all of these boundary proteins that we talked about. We talked about heterochromatin and euchromatin back in previous parts of the course. So I like this as a connection to some of those previous parts of the course here. So moving on a little bit to just thinking at a bigger sense rather than just looking at these individual examples. We talked a little bit about feedback loops and the circuit diagram of thinking about how gene regulation takes place. And one of the best understood of these, and one of the earliest ones to be understood, were the positive feedback loops that had to do with the so-called genetic switch in lambda bacteriophage. And the genetic switch, which is diagrammed up here, is the decision whether a virus, when it's infected a cell, is going to blast open the cell and make a whole bunch more virus, or it's going to integrate its genome into the host genome and just hang out. So once that decision has been made, the cell needs to remember, and this is the cell with the virus in it, whether it's going to hang out or whether it's going to make a whole bunch of viral genes. And so once the decision has been made, how the decision is made, lecture 5, I think, in virology next term. But once that decision has been made, you either are making the lambda repressor, which, again, repressor should be in quotes because it represses some genes but activates others. And in this case, it activates the expression of its own gene. So that's your positive feedback loop in that case. So if you decide, or the cell decides, that it's going to be integrating its genome and hanging out, this is what happens. If the other decision is made, where you have now turning off of this gene, but turning off of our friend CRO. Does CRO sound familiar? That dimer with the 2 beta strands binding to DNA? So that's exactly this protein. It serves as a positive regulator in the opposite way. It's now expressing other genes, which are now going to lead to massive production of all of these virion genes and the cell being completely blown apart. So 2 different positive feedback loops which happen there. Now, we also talked about the negative feedback loops where you have repressors, which are going to repress the transcription and expression of other proteins. That can give you an oscillating cycle. You can also have the 2 activators that are acting sequentially, which lead to the filter, as again, sort of an electric filter, where you can think about this being a timing device to say, as long as we have a signal that lasts for long enough, then we can turn on a second signal. Yeah, Olivia.
So the lambda crow, is it an activator for its own?
So take the virology course next term. We'll talk a little bit more about it. But so the question is CRO also an activator for its own gene? It turns out it allows other activators to bind. So it's not, in fact, doing that. It's a repressor for the C1 gene, or the lambda repressor gene.
But it's still a positive feedback loop in terms of when the protein is translated or transcribed, it needs more lambda crow.
Well, it will allow more lambda crow to be made. And so it's not like the lambda repressor, which actually is both an activator and a repressor. CRO just serves as a repressor. But there are other proteins in that, because again, we're oversimplifying the system. And so there are other proteins which will lead to that expression. Yeah?
The
It's blocking CRO, exactly. Yep. It's activating its own gene, exactly. Okay, on the other side, we have gene silencing. Also, getting back to what happens with yeast mating type switching. What happens in silenced genes? You have first modification of the histones. Modification of histones usually in terms of methylating those histones. After you have histone methylation, you will have these proteins and the read-writer complexes that will bind to methylated histones and then methylate other histones which are next to them. Now you have a whole stretch of methylated histones. Then you will have a DNA methylase, which will methylate the DNA. Then you have a DNA binding protein that only binds to methylated DNA, which will also bind to the DNA. And so it's this whole process: histone modification, DNA modification, and extra binding proteins that really gives you this massive silencing. So going from some expression to absolutely none, well, absolutely none is 1,000,000 times less, which, as far as we're concerned, is none. So you can have this massive amount of change of expression of one particular gene can be, under one situation, 1,000,000 copies of your gene, and in another situation, 1 copy of that particular gene. But it's modification of histones, modification of DNA, and all of the binding proteins that are associated with it. So it's that whole process. And the X chromosome inactivation is also very similar. So you have modifications of histones, modification of DNA, and all these proteins, which will also help do this compaction. DNA methylation is not just for compaction of DNA. We also have specific methylation, which will happen, again, at very specific sequences, not just the general case here. Mostly seems to be important for gene imprinting, where you can have DNA methylation that happens either in maternal or paternal chromosomes, and that process will block the binding of particular proteins. And again, sequence specific now, not the euchromatin is going to be sequence non-specific. Here, specific binding, which will block some kind of DNA binding protein. That could be an activator, could be a repressor. In the case of IGF-2, it actually was binding of an insulator element, which will block the activity of an enhancer from somewhere else in the genome. That's transcriptional regulation. Post-transcriptional regulation, we talked about a number of different things, but I did want to specifically mention RNA editing, because I think I did a really crummy job of explaining this the first time around, so I wanted to give myself a second chance. The ADAR enzyme. So ADAR stands for adenine deamination acting on RNA. So went back and looked that up. So adenine deamination goes from adenine to inosine, which is going to change the base pairing interactions that happen at that particular nucleotide. This only happens if you have a double-stranded RNA sequence that the ADAR enzyme is going to interact with. Because if you modified all the adenines, it would be a god-awful mess. So this happens in only a few cases, and it turns out to be absolutely critical for brain development. If you don't have this particular, and this is one of the ADAR genes, there are quite a few of them, but one particular ADAR gene, mice die before they're actually ever even born. And why you have to change these nucleotides and where that comes from, ask Mr. Darwin, because there's no particular reason why you shouldn't just mutate this and have a different gene there. So, but these are the things that happen. Again, in virology, we'll talk about some of the really fascinating ways that viruses can avoid this problem. One of the other sets of things that ADARs do is they'll modify RNAs in viral genomes such that those genomes are mutated so much that they can't replicate anymore. But then viruses have evolved ways to deal with this as well. Another thing I did, I think, not a good job explaining was having to do with RNA localization. Why do you have bicoid and hunchback only in particular parts of your Drosophila embryo? That's because the messenger RNA for bicoid and hunchback is put at one particular place in the egg by the egg development in the mother of that particular egg. And so this is maternal messenger RNA that gets put at one end of the embryo. How does it get put there? It gets put there through binding proteins to the untranslated regions in your messenger RNA. So we talked about the 5' end. We saw that right at the beginning. 3' end has proteins that will bind to this and then get that RNA to a particular place in the cell where it turns out to be used, and you have these gradients, and you can get even skipped activation appropriately. These get moved around in the cytoskeleton, and just where you happen to need an RNA can be very, very useful in terms of the regulation of actually getting your particular protein. Multiple different ways how this happens. Let's not worry too much about it. The main thing here has to do with these untranslated regions and specific binding proteins that will associate with them. We spoke briefly about translational regulation. Most of this has to do with the stability of your messenger RNA or initiation of translation. Initiation of translation, EAF2, binding to, and again, this is nice, how I'm going to have things on the previous sets of exams on this exam. EAF2, we learned about it last time. Talk about its regulation now. So the regulation of EIF2 has to do with GTP binding and the recycling of the guanosine triphosphate. If you don't have recycling of your guanosine triphosphate, then you're not going to get translational initiation. This turns out to be a really nice way of the cell to respond to a nasty virus infection, for instance, saying, no, I don't want to translate any of your nasty viral genes. I'm going to turn off translation completely. Or if you're going into a part of the cell cycle where you don't want to be translating too many genes, a really easy way to regulate that you're not going to be translating very many genes is just to stop translating them. And the way to do that is phosphorylation of EAF2. And again, we talked about RNA stability. Messenger RNA stability mostly has to do with the two ends of your messenger RNA being associated with each other. The 3' end, which has your poly A tail, the 5' end that has the cap, and then often other proteins that are going to be associated with either the 3' UTR or the 5' UTR, which will block the hungry exonucleases that otherwise would be chewing up your messenger RNA. And so the presence or absence of some of these proteins, we looked at the aconitase example, has a lot to do with the stability of a messenger RNA and how much of it you get back out. Quickly go over some of what pre-Dr. Kaysen talked about in his lecture. Long non-coding RNAs. There are many, and even more of these that are being discovered. These long non-coding RNAs can be involved in transcription of particular genes, and this is the activity in cis. So we have a long non-coding RNA which is being made, proteins associated with that long non-coding RNA, and block transcription in a region right next to that long non-coding RNA. But they could also act in trans, where we have, again, proteins that bind to a long non-coding RNA that have some kind of activation at a distance. This can be activation, it can be repression. We're still learning a lot about how these long non-coding RNAs are functioning. The shorter RNAs that George talked about are really in two classes. There's a third one that we didn't talk about. microRNAs and small interfering RNAs. MicroRNAs are the endogenous, so they're present inside the cell. There are genes that express these short RNA sequences. These are then processed, they get capped, they get tailed, and then they get chopped into smaller pieces. And these smaller pieces, this is going to be your microRNA that gets made, gets chopped by dicer into a shorter piece of RNA that can either lead to RNA degradation or it can lead to suppression of translation, again, usually by binding in these UTR sequences through base pair interactions. Really long base pair interactions lead to cleavage of your RNA, the messenger RNA. Short ones usually lead to suppression of translation. Small interfering RNAs, these are usually exogenous RNAs, so it'll be a viral DNA which has come inside the cell, or in some cases this will be random RNAs which are being transcribed from the genome, any of these copied RNAs. If you've got many, many copies of a particular kind of RNA, those are going to be all these repeated elements, like all those retrotransposons that we talked about again way back when we talked about the structure of the genome. 40% of our genome are these retrotransposons. So you'll have a double-stranded piece of RNA. That double-stranded piece of RNA gets cut again into smaller pieces. And again, if it's got a perfect match to another RNA which is there, which would make a lot of sense if it's a viral RNA, that will get chopped up through the RISC complex, which is just like what you see with microRNAs, or in the case of these endogenous sequences, usually the transposons, that RNA is used in a transcriptional silencing, and that transcriptional silencing is through DNA methylation, histone methylation, the whole silencing process which we just talked about. Most of George's lecture, he talked about CRISPRs, the clusters of regularly interspersed palindromic repeats. These are DNA, which provide defense against viral invaders in bacteria and archaea, but have now been used for genome engineering in eukaryotic cells. So the process, you have a virus which releases its DNA inside the cell. We have the adaptation response where the bacteria will take this piece of DNA, add it to your CRISPR array between all of these repeated sequences, these spacers. Once you have a spacer, that can be transcribed, and any future infection will be blocked because you have one of these RNAs which will block the DNA once it comes inside the cell. There are 3 steps: adaptation, expression, and interference. The adaptation step, I'll show you maybe back up here. Here we have adaptation, expression, interference. How does it happen? Particularly in the type 2 systems. The type 2 are the easy ones, which is why this is what's being used for genome engineering, having to do with Cas9. Adaptation is Cas9 plus Cas1 and Cas2. These are the CRISPR-associated sequences, which will bind to that foreign DNA and put it into your CRISPR array. So this is where you're figuring out what you've been infected by. Then that CRISPR RNA locus, which is the regularly interspersed palindromic repeats, and again, a palindrome is sequences which are now base pair with each other. Read one way, read the other way gives you the same sequence, which means that they can base pair with each other. These get made into RNAs. Because of this palindromic sequence, they get chopped into smaller pieces, and those smaller pieces are going to be what interacts with your Cas9. And Cas9, once it has one of these shorter sequences associated with it, if there's now a new infection that happens with DNA, happens also with RNA in some of these CRISPR systems, it can now chop that incoming DNA, again, or RNA, but usually DNA, at a very specific position with the 2 different nuclease domains that are present in part of Cas9. Once you have that, you have a double-stranded break, and this is how all of the genome engineering is done. You have a RNA sequence which matches what you want to get rid of in your genome. You put it together with Cas9, you get all of that inside a cell, and it chops up the DNA where you want it to be chopped up. That can either be a double-stranded break that gets repaired by non-homologous end joining, which chews in at ends and makes a mutation, usually a deletion, or you can put in a homologous DNA and through homologous recombination replace whatever it was that you cut in the first place and are now being repaired. So I'm not going to talk about methods. We didn't get there, and I didn't give you 5 more minutes to fill out your course evaluations. But if you have a few more, please do that. It's useful to me. I have tenure and I'm a professor, so you can't fire me through this. But they could give me a hard time. So please let me know what you think about this course. Thank you for a fun term. We'll see you on Tuesday morning.